A very warm good morning, everyone. So, hope the audio and video is perfectly clear. Yes. So, once again, we are back from the Team by Point Stream, the world of bio, with yet another session on Cell, the unit of life, the lecture one. Yes, everyone. Yes, this we need 2023 batch. So, once again, we are going to start the new chapter, Cell, the unit of life. Yes. So, hope the audio and video is perfectly fine. Yes. So we are moving on to that without any further delays. So this is entirely a different chapter that is a different unit that is cell the unit of life, biomolecules, cell cycle and cell division. These are the three different units. Yeah, three different chapters in this unit. Yes, everyone. So we are moving on with today's session. Before moving on to that, reminding you to join the Telegram group where you have daily session updates, regular discussion, where you have daily polls and you can interact with me. And the direct link to join the Telegram group is given there in the description box, that is t.me/neat_biopoint. Yes, everyone. So without wasting much time, we are moving forward. Yes, we are analyzing as I'm doing in each and every chapter in the first session of my chapters. Yes, let's discuss the weightage of the topics of this chapter. So different topics of this chapter are cell theory, an overview of cell, prokaryotic cell, eukaryotic cell, plant cell and animal cell, cell envelope, cell organelles. So these are the different topics of this chapter. Yes, and as you can see, the 69% weightage of this chapter goes to cell organelles. 69% weightage. Yes, and cell envelope, that is very minute. You cannot even see that. Okay, that's much very minute. Plant cell and animal cell, that is 3%. Cell theory 2%. Overview of a cell, all these are does not given here because that all are very minute topics. Okay, prokaryotic cell 23% and 3% eukaryotic cell introduction. Okay, so that about the last previous year 8 years weightage. And if you are analyzing 33 years NEET or IIPMT weightage, you can see 67% cell organelles, 10% cell envelope, 1% plant cell and animal cell, 2% eukaryotic cell, 17% prokaryotic cell, 1% overview of a cell, and 2% cell theory. This is what the previous 33 years analysis, weightage analysis of NEET or IIPMT on the subtopics of this chapter, cell, the unit of life. Yes. So without wasting much time, we are moving forward with today's session. Yes, do hit the like button, do share it with your friends, and do subscribe by point stream the world of bio. Yes, we are moving. Hope you all are ready. If it's fine, give me thumbs up on the screen. The very first slide of the day. What is a cell? Yes. So, what is a cell? A cell is the fundamental, structural, and functional unit of all living organisms. Yes, Binaya, Paki, Abhijit. Yes, I forgot to read all your names. Calvin, Yamini. Yes, everyone. So, the very basic fundamental unit of life is known as a cell. For example, if you're saying about the fundamental unit of nervous system, the fundamental unit of nervous system, it is what? A nerve cell or a neuron, right? Neuron. Yes, Krishna and Yadav. Krishna and Yadav. Sorry, I just read it as Krishna and Yadav. Very sorry. Okay. So as we are discussing, the basic unit of nervous system as neuron, the basic unit of excretory system, or the basic or fundamental unit of kidneys as nephron. Nephron. In this same way, the basic or the fundamental unit of life is what? It is the cell. So the fundamental structural and functional unit of all living organisms is known as cell. Yes. So who discovered this cell? cell is discovered by a scientist known as Robert Hooke. So cork cells, which cells was discovered by him? Robert Hooke discovered cork cells. All these are very basic introduction that you have already learned in your class 9. Yes, and the very first observation of a living cell. Okay, first living cell was, yes, Paki. It was in 860. Oh my God, it was in 1665 that Robert Hooke discovered the very first cell, that is cork cell, and the first one to observe and discover a living cell was Anton van Leeuwenhoek. Yes, first one to observe living cell was Anton van Leeuwenhoek. Yes, everyone. Okay. The invention of compound and electron microscopes revealed the structural details of the cell. So these were done by all these scientists. Just did the basic invention, basic discoveries by the help of a simple microscope, and later this for the further detailed structure of all these cell structures, a microscope was discovered, that is compound microscopes and electron microscopes. Yes, everyone. So that each and every minute details of this can be obtained. Yes. So Robert Hooke discovered dead cells in the cork from the plant cork, and then van Leeuwenhoek observed the living cells, that is from RBC and sperm cells. Yes, everyone. RBC and living cells. Oh my God. RBC and living sperm cells was the cells that Anton van Leeuwenhoek used for discovering cells. Yes. Next, we are moving on with the cell theory. That is, first, a scientist called Matthias Jacob Schilden, or Matthias Schilden, in 1838 composed that plants are composed of cells. Okay, and in 1839, another scientist called Theodor Schwann said that cells had a thin outer layer called plasma membrane, and he also found that plant cells have cell wall. And he proposed the hypothesis that animals and plants are composed of cells and products of cells. Yes, yes, Paki, Binaya, and Yamni. Abhijit, are you here? Calvin, are you here? Yes. So Matthias Jacob Schilden, as we have discussed, is a botanist. That is about, he studies about plants. So he's a botanist. And Theodor Schwann was a zoologist that is studied about animals and said that animals cells composed of an outer layer called as plasma membrane. Yes. So, and finally, they concluded that animals and plants are composed of cells and products of their cells. Okay, this is what the very basic cell theory. So, if anyone is asked, or if a question is asked, who proposed the cell theory or who formulated cell theory, it was done by Schilden and Schwann. Yes, Schilden and Schwann formulated the cell theory. And who put forward the modern form of cell theory? It was done by another scientist called Rudolf Virchow. Yes, it was done by another scientist called Rudolf Virchow. Yes, everyone. So we are gonna move in. We are gonna learn detail about each and every points of that. Okay, so Rudolf Virchow explained that cells divided and new cells are formed from pre-existing cells. That is, new cells arise from the pre-existing cells that are already existing. Okay, so he just said a term called omnicellular e-cellular. Okay, the term that was said by Rudolf Virchow was omnicellular e-cellular. What does that mean? The term omnicellular e-cellular means that all cells are arising from the pre-existing cells. Yes, Calvin and Paki. So that refers to that all cells are arising from pre-existing cells. So that way, in the same way, the modern form of cell theory was formed, and that was put forward by Rudolf Virchow along with the points or the points formulated or contributed by Sheldon and Schwann, and that is all living organisms are composed of cells and products of cells, and all cells arise from pre-existing cells. This is about the modern cell theory. Yes, everyone. This is a NEET 2019 question. Yes, a NEET 2019 question about the cell theory. Okay, so next we are moving on with the next topic, that is an overview of a cell. We have to complete prokaryotic cell also today. So we have almost 27 slides in total. So if we strive hard, we will be able to do that. Okay, so all cells contain cytoplasm. What is the cytoplasm? It is a semi-fluid matrix where cellular activities and chemical reactions occur. That is, this keeps the cell in living state. Yes, this is what keeping the cell in living state. So all the cell organelles, that is mitochondria, ribosomes, for the endoplasmic reticulum, Golgi bodies, all these are just located within this cytoplasm. So all the cellular activities, cellular metabolism, all these are occurring within this cytoplasm, and this is what making the cell in a living state. Yes, Paki, it is a jelly-like structure. That is what we have said by semi-fluid matrix. Okay, semi-fluid matrix. Then ribosomes, that is, they are non-membrane bound organelles found in cytoplasm. Ribosomes are non-membranous. There is no specific membrane bound covering around them. Okay, that is found in cytoplasm, chloroplast, mitochondria, on rough endoplasmic reticulum. All these structures or all these organelles, these ribosomes are found. That is within this cytoplasm, in the chloroplast, mitochondria, and rough endoplasmic reticulum. Endoplasmic reticulum with the presence of ribosomes is known as rough endoplasmic reticulum. Okay, everyone. So, endoplasmic reticulum with the presence of ribosomes is known as rough endoplasmic reticulum. So, that's all about an overview of a cell. Then we are going to learn different shapes and size of cell. Smallest living cells are mycoplasma. Yes, we have discussed that in biological classification. So, what is this mycoplasma? What is this mycoplasma? Mycoplasma is the smallest living cell without a cell wall. Smallest living cell or living cells that are lacking cell wall is referred to as what? Mycoplasma. And second point, largest isolated single cell is egg of ostrich. Egg of ostrich is a single cell. This entire egg of ostrich is a single cell, which is the largest cell. Okay, largest one. Longest cell is nerve cell. The longest cell is the nerve cell, and the size of bacteria is 3 to 5 micrometer, and human RBCs are 7.0 to 7.0 micrometer in diameter. Mycoplasma is found in which organism? Mycoplasma itself is an organism. Yes, it is a living organism. Mycoplasma is a living organism. Okay, and that is a living organism without a cell wall. Smallest living organism without a cell wall. Yes, yes, it can survive without oxygen as well. Yes, that too is a point to be added. Then, based on the functions of cells, shape of cells may be disc-like, polygonal, columnar, cuboid, thread-like, or irregular. And cells on this cells are of two types, that is prokaryotic cells and eukaryotic cells. What is the difference between prokaryotic and eukaryotic cells? Yes, prokaryotic cells refers to that unicellular group of cells. Okay, unicellular group of cells is known as prokaryotic cells. That is without, without a membrane. Okay, non-membranous cells are known as prokaryotic cells. Okay, their nucleus will not be having a membrane, and that is what is said to be a prokaryotic cell, whereas eukaryotic cells have a true nucleus with a well covered nucleus. Okay, well, membranous covered nucleus. Yes, everyone. So now we are gonna move in detail about prokaryotic cells. Okay, is it clear till now? Okay, so prokaryotic cells have no membrane bound nucleus and organelles. Yes, no membrane bound nucleus and organelles are present in case of prokaryotic cells. Okay. They include bacteria, blue-green algae, mycoplasma, and PPLO, that is pleuropneumonia-like organism. We have said that mycoplasma is also known as what? PPLO. Yes, they are generally smaller and multiply more rapidly than eukaryotic cell, vary in shape and size. This is a very basic introduction. So, what is prokaryotic cells? They are not having membrane-bound nucleus or organelles. And examples include bacteria, blue-green algae. What is blue-green algae? It is also known as cyanobacteria. Yes, we have learned in biological classification what is this blue-green algae. That is cyanobacteria. Mycoplasma, PPLO, that is pleuropneumonia-like organisms. Yes, everyone. Bacteria have four basic shapes, that is Bacillus, Coccus, Vibrio, Spirillum. Bacillus is a rod-like shaped bacteria. Coccus is the spherical shaped bacteria. Vibrio is the comma shaped bacteria, and Spirillum is the spiral basic shapes of bacteria. Bacillus, Coccus, Vibrio, and Spirillum. Yes, everyone. So hope it's clear till now. I know I'm moving a bit fast. Is it okay? Are you able to follow? All these are basic topics that we have covered in lecture 9. Lecture for class 9. Yes, yes, Yamni, Calvin, Paki, Abhijit. Okay, Benaya. Okay, fine. So we are moving on with the cell organelles. Okay, so cell organelles, which all are the organelles that we have to cover in this topic? This session. Okay, the very first organelle is this. Very first topic is the cell envelopes. First, we have to cover about the cell envelopes. Second, mesosomes and chromatophores. Third, nucleoid, excuse me. Fourth, flagella. Fifth, pili and fimbriae. Sixth, ribosomes. And seventh, inclusion bodies. So these are the very sorry. These are the different cell organelles that we have to cover in this session. Okay. So, cell envelopes. First, we are going to say about the cell envelope. And cell envelope is a chemically complex protective covering. A cell, if this is a cell, it will be having a protective covering outside that. And that is what referred to as an envelope, an outer covering. Yes, for example, when we are writing a letter itself, we will be having no, never. It's not COVID symptoms. Okay, something is burning there, and that's why the smell of that. Okay, okay. Chemically complex protective covering. That is, when we are just posting a letter itself, we will be having a postcard cover, right? As a letter cover, and that is what set as a cell envelope. That is what set as an envelope. In the same way, the such membrane or a structure that is covering a cell is referred to as a cell envelope. Yes, and it is made of three tightly bound layers, and that is glycocalyx, cell wall, plasma membrane. Yes, glycocalyx, cell wall, and plasma membrane are the three different cellular layers. Membranous cellular layers. Okay. Yes, the outer covering, three different outer covering is glycocalyx, cell wall, and plasma membrane. The outer glycocalyx, middle cell wall, and inner plasma membrane. Yes, this is all about the basic introduction of cell envelope. Now we are going to learn all this in detail. Yes, what is this glycocalyx? That's what my question is. Glycocalyx is the very outer layer of what? very outer layer of the cell. Very outer protective layer of the cell is known as glycocalyx. Its composition and thickness vary in different types of bacteria, and it may be slimy layer or capsule layer. If the glycocalyx is loose, if the glycocalyx is loose and thin, it is known as slime layer. Slime layer. And if the glycocalyx is thick and tough, it is known as capsulated layer or capsule. Okay, loose and thin it will be slime layer, and thick and tough it is capsule. Okay, this is about glycocalyx, a basic introduction that all about glycocalyx. Cell envelopes can be divided into three: glycocalyx, cell wall, and plasma membrane or cell membrane. And this glycocalyx is the outermost layer, and it is of two types, that is slime layer and capsule layer. That is, if the glycocalyx is loose and thin it is slime, and if it is thick and tough it is capsule. Okay, is it clear? Is it clear? Next is all about cell wall. Cell wall is the middle layer, and it is seen in case of all prokaryotes. Okay, and it gives shape to cell and provides structural support. Yes, Yamini, Calvin, Paki, Vinaya, Abhijit, where are you? Okay, so middle layer seen in all prokaryotes, that is cell wall. And what is its function? Its function is to provide shape to the cell and provide structural support. Why it is providing that? Why the cell wall is essential? In order to prevent the bacterium from bursting or collapsing. That is one of the main functions. That is, it provides a structural support in order to prevent the collapsing of the entire bacteria. Okay, Abhijit. Okay, in order to prevent collapsing of the bacteria, the cell structural support, the cell wall is providing structural support and a definite shape to the bacterial cell. Yes, everyone. Is it clear? I haven't received any thumbs up yet. You're commenting yes, but give me thumbs up. That's what I asked. Yes. So that we can move with the next topic, that is the cell membrane. Yes. Come on. Okay. So hope the glycocalyx is clear. Cell wall is clear. Okay, fine. Next is plasma membrane. Yes, inner layer. Plasma membrane is the innermost layer of a cell. For example, if we are having, if this is a cell, we will be having a first covering, that is looking from inner to outside. The first covering, plasma membrane. Then you have a covering above that, that is cell wall, and more above that, that is the glycocalyx. A cell is located inside these three different types of cell envelope: glycocalyx, cell wall, and plasma membrane. Yes, everyone. And it is semi-permeable in nature and interacts with the outside. Plasma membrane is semi-permeable in nature. This is structurally similar to that of the eukaryotes. Plasma membrane is structurally similar to that of the eukaryotes. All the eukaryotic and prokaryotic organisms have this innermost layer called plasma membrane, and their structure is same in case of both eukaryotic and prokaryotic organisms. Yes, everyone. Yes, come in me. Yes. So the innermost layer is the plasma membrane. It is semi-permeable in nature. So what is the semi-permeable? That is, it allows only certain substances to enter and leave the cell. Only permit certain substances to enter and leave the cell. Okay. So plasma membrane is made of lipids and proteins. Yes, plasma membrane or cell membrane is made up of lipids and proteins, the same as that of the composition in eukaryotic organisms. Yes, everyone. Lipids and proteins is the composition of cell membrane. Yes. Okay, so based on the types of cell envelopes and response to gram staining, what is this gram staining? It is a method, or it is the type of method. I mean, it is a type of staining developed by a scientist called Christian Gram. Yes, a scientist called Christian Gram developed this method of gram staining. On that, different types of stains are added to the bacteria, differentiating these types of bacteria mainly into two as gram positive and gram negative bacteria. So, what is this gram positive and gram negative bacteria? Yes, when the stain is poured onto the bacteria, if they retain the gram stain, if they are still retaining the gram stain, then that type of bacteria will be classified under gram positive type of bacteria. If they are not retaining that stain, then it is known as gram negative bacteria. What is this gram positive and gram negative? What is this gram? Gram is named after a scientist called Christian Gram. Yes, I will repeat that. What about this gram staining? All about this gram staining. It is a method of staining. What is this staining? Yes, what is this gram staining? It is a common method that is used to differentiate between two different types of bacteria. Okay, different methods. I mean, a common method to differentiate between two different types of bacteria. That is by pouring certain stains. What is the stain? The stains are certain colors. Okay, colored dyes. Okay, certain color dyes will be formed, I mean, poured onto this bacteria, and some bacteria will retain this color, keep on that, stick on to that color, and some will not absorb, I mean, will not stick on to that color. Okay, if they are just retaining that stain, then it is known as gram positive bacteria, and if they are not retaining the stain, then it is known as gram negative bacteria. Is it clear? Is it clear about gram positive and gram staining, Abhijit? Yes, come on. One, two, three, four, five, six, seven, eight. Eight more slides left. Okay, fine. Is it clear, everyone? Come on. Okay, fine. Okay, I haven't received. Yeah, it was hang. The chat was just hang. Okay, that's why. So it is used to find whether glycocalyx is present or not. No, no, no. It is used to classify two different types of bacteria. That is whether it is large or small. Two different types of large bacteria or two different types of small bacteria. All these are just differentiated or just classified on the basis of this gram staining method. Okay, that is stains are poured onto that, and whether if it is just a what keep on sticking onto that stain, then we will say that gram it is positive gram stain, I'm gram positive, and if it is not retaining, then we will say that it is gram negative. So if gram staining is not possible, then what criteria is used? Gram staining, it is not a criteria. If you are saying about gram staining, it is just a method. That is, we are just learned a method whether gram staining is there or not. Okay, that is gram staining. If whether to distinguish between a large cell or a large bacterial cell or a small bacterial cell, a method is possible, and if that method is possible, and that is gram staining. Okay, we have just learned, or we have just gave a brief introduction onto that about what is this process of gram staining and whether it is applicable in case of this prokaryotes or not. Okay, whether this gram staining method is applicable in case of this prokaryotes or not was just we have analyzed now. Okay, is it clear? Okay, fine. So next organelle or next structure that we are going to learn is Mesosome. Hope it's a new term. Mesosome and chromatophores. Hope this is a new term for all of you. Okay. So what is this mesosome? It is formed by infolding of plasma membrane. All of you know that. Hmm, how could I draw? Please wait. If you are taking this as a bacterial cell or a cell that we are going to analyze the right now, that is comprising of, oh my god, that is comprising of three outer layers. Yes, three outer layers. Yes, that is plasma membrane, cell wall, and glycocalyx. Yes, plasma membrane, cell wall, and glycocalyx. Yes, everyone. Plasma membrane, cell wall, and glycocalyx. Okay. Plasma membrane, cell wall, and cell glycocalyx is the three outer or the cell envelope, and from that inside the cell membrane, what is the cell membrane, also known as plasma membrane? There will be an infolding. Okay, there will be an infolding in case of this plasma membrane. So if you are learning this as this plasma membrane, there will be an infolding regarding that. Okay, an infolding will be there, and that is known as, look here, and that is known as mesosome. This infolding is what known as mesosome. If you look in this diagram, this plasma membrane is just infolded, and that is what known as mesosome. Yes, yes. Okay, so pigmented mesosomes, that is, if pigments are present in case of this mesosome, then it is known as chromatophores. Pigmented mesosomes are known as what? Chromatophores. Yes, everyone. So hope it's clear about mesosomes and chromatophores. Mesosomes are the infoldings formed by plasma membrane, and pigmented mesosomes is known as chromatophores. Okay, and mesosome includes vesicles, tubules, and lamellae. It includes vesicles, tubules, and lamellae. These are the components present inside a mesosome. Yes, all these help in cell wall formation. Formation of cell wall is just helped by these components, vesicle, tubules, and lamellae present inside a mesosome. It also helps in DNA replication. Yes, helps in the replication of DNA. Okay, so this helps in cell wall formation, replication of DNA, and distribution of this replicated DNA to the daughter cells. Distribution of this replicated DNA to the daughter cells. Okay, is it fine? Okay, okay. So hope it's fine. So Abhijitas, what is plasmid? Plasmid is an extra chromosomal material in a prokaryotic cell. It is an extra chromosomal material in a prokaryotic cell. And let me check whether we have to learn it in the further slides. No. Okay. So, as we have discussed the function of mesosome, that is for cell wall formation, for DNA or chromosomal replication, distribution of chromosomes to daughter cells, respiration and secretion, that is helps in the cell respiration of the cell and secretion, increases the surface area of plasma membrane and enzymatic content. For example, if a line is moving this much, oh my god, if a line is having this much length, okay, and another line with this much length, which is of, I mean, another line in this shape and first line in this shape, which is having more space or more length. Line 1 or line 2 is having more length. Come on, come in me. Line 1 or line 2 is having more length. Yes. Second one, yes, because if we stretch the second one, this extra length also will get added, and that helps in the elongation of the second line too, right? So in the same way, if there is an infolding in case of this plasma membrane, that helps in increasing the surface area of the plasma membrane. Yes. Okay. Chromatophores. Chromatophores are pigment containing membranous infoldings in some prokaryote. Yes. Pigmented mesosomes is known as what? Chromatophores. Example, cyanobacteria. Cyanobacteria also known as what? Blue-green algae. Blue-green algae. Yes. Pigmented mesosomes is what known as chromatophores. Yes. Next is about nucleoid. Okay, so from nucleoid we will discuss in the very next lecture. We are stopping till mesosomes. So what I am informing you is that I hope today night by 8 p.m. all of you are free. Is it? Are you all free by that time, 8 p.m.? Yes. Okay. So we will conduct a telegram quiz regarding the topics that we have completed till today. I know I have moved a bit faster. Okay, I hope each and every point I have completed till today is perfectly fine. Okay, so that's all for today. Reminding you to join the Telegram group where you have the daily session updates, regular discussion, daily polls, and you can interact with me. The direct link to join the Telegram group is given there in the description box. Okay, and that is t.me/neat_biopoint. Yes, it's a easy chapter. That's why I'm not able to explain much on these topics. But when you are reaching eukaryotic cells, I will be able to do that. Some more detailed topics is there. Okay, so it's not because of my issue to explain, it's the issue that the chapter or the each and every point of that we have completed today is able to understand just by reading. Okay, so again reminding you, do hit the like button, share it with your friends and subscribe biopoint, stream the world of bio. And that's all for today. We will be coming with the lecture 2, sell the unit of life, very soon. So till then we meet. So please today give PPT. You mean the PPT of the slides that we have completed till now? Yes, if it's today we will do it. Okay, as per your request we will do it. Yes, everyone. So thank you all. Thanks for your patient listening. It's me signing out from BioPoint, stream the world of bio. Bye bye. So all of you stay ready for the Telegram quiz at 8 p.m. Okay, the further instructions, I mean reminder, will be given in the group itself. Okay, bye bye, guys.